All right, so we're working on the grading of the exams. We plan to have those back in the boxes by Wednesday morning. So when you come to class on Wednesday, you'll be able to pick those up. The answer key will be posted, and we'll talk about it then. Today we'll continue our discussion of interesting compounds. We talked about alcohols on Friday. Today we'll talk about ethers and epoxides, spending more of our time on a class of ethers called crown ethers. We'll talk about how alcohols, ethers, and epoxides are synthesized. And the main reaction we'll focus on there is called the Williamson ether synthesis. And then we will talk about, we'll start talking about the reactions that alcohols, ethers, and epoxides undergo. And we'll begin with one called dehydration. So we learned about an interesting ether back in chapter 3 when we talked about monensin. Ionophores are usually ethers. Ionophores are molecules, organic molecules that coordinate to cations. And we showed you in chapter 3 how monensin uses ion dipole interactions between those Lewis basic oxygens and the silver cation to complex it, to hold it in this cavity. And then it's able to transport that cation across the cell membranes, causing cell death. There are synthetic ionophores, and the structures are much simpler than monensin and the other ionophores from nature. And those synthetic ionophores are known as crown ethers. And the most common crown ether is the one I've drawn here on the board, and it's called 18 crown 6. So the nomenclature for crown ethers involves the first number telling you how many atoms there are in the ring. So this is an 18-membered ring. And then the 6 is telling you how many of those atoms are oxygens. So an 18-membered ring with 6 oxygens, and they're spaced out evenly. We've got CH2, CH2 groups in between each of our oxygens. Okay, so that's the simple nomenclature associated with crown ethers. So here we have on the left an electrostatic potential map of 18 crown 6. And we see this cavity in the middle, this highly Lewis basic cavity, where we have all of the oxygens, all their lone pairs pointing into that cavity. So we have a lot of electron density there. So it's tailor-made for coordinating to a cation. And on the right, what we see is a space-filling model where this big lavender-colored or purplish lavender-colored sphere is a potassium cation. That cavity is the right size for a potassium cation to fit very snugly. Sodium cations will also fit reasonably well, but potassium fits exceptionally well in the cavity of 18 crown 6. So this is not just a chemical curiosity or just something that's interesting. It is interesting, but it's interesting and useful. There are lots of applications for crown ethers. Before we talk about them, I should mention, so the crown part of the name just comes from the fact that the structure kind of looks like a crown that you could wear on your head if you made a model of one and you had a big enough model set. So that's where the name comes from. So on this slide, we see the application that is most relevant to our class because it talks about something we just learned about, namely SN2 reactions. So you can take a salt. Remember, most of our nucleophiles are anions, and they are usually used as salts. There's a cation that's a spectator, but they don't dissolve in nonpolar solvents. So that's why we have said that up until now, you have to use polar solvents to perform an SN2 reaction. But when you use a nonpolar solvent and a crown ether, what happens is you form this Lewis acid, Lewis base complex, which you can also call a host-guest complex, where the crown ether is the host and the potassium cation is the guest. This is a cationic complex, but it will dissolve in nonpolar solvents because it has all of these CH2 groups. It has sufficient nonpolar regions in the molecule that it will dissolve in a nonpolar solvent, such as benzene. This is benzene written right here, but it could be any nonpolar solvent, and it will dissolve. But it has to bring its anion with it. You can't just have the cation without the anion. So the anion comes with it in solution, but it's floating around with no intermolecular attractive forces. The cation it would be attracted to is attracted to our crown ether. There's no ability to have ion dipole interactions with our benzene solvent, so we have a naked nucleophile, free of intermolecular forces, highly reactive, even more reactive than it would be in a polar aprotic solvent. So we can have an SN2 reaction that would be even faster than what we would see in polar aprotic solvents such as DMF or DMSO. Okay, so here, this rapid SN2 reaction will occur using the combination of a nonpolar solvent and a crown ether. Any questions? So crown ethers have some history with BYU. There was a longtime organic chemist named Gerald Bradshaw, who was a professor here at BYU from the 70s until about 2000. And Professor Bradshaw's research focused on crown ethers. He synthesized crown ethers and studied a lot of their properties. He co-founded a company based on crown ethers. So he got a lot of mileage out of his crown ether research. And I actually performed undergraduate research in his lab back in the 90s when I was an undergraduate here and made some crown ethers myself. But Professor Bradshaw loved crown ethers so much that he had a personalized license plate based on crown ethers. 18 crown 6 would not fit on his license plate, so he had crown 6. And unfortunately, hardly anybody knew what it meant. Most people, when they saw his license plate, thought he was a dentist or something like that because of the reference to crowns. But some of us knew the meaning of that license plate that he had. So after he retired, he and his wife served a mission. They went to Jordan, a very interesting place to serve a mission. When they came back, I asked him if he still had his crown 6 license plate. And he said that he did not, because while he was on his mission and his car registration expired, somebody else had snagged that license plate. They had asked for crown 6 as a personalized license plate, and they had it, and so he was unable to get it back when he returned from his mission. So if you see someone driving around with a crown 6 personalized license plate, then you can give them a dirty look because they're the person that stole this license plate from Gerald Bradshaw. They probably had no idea what the meaning was. I'm not sure why they would use it, but anyway, so crown ether research has gone on at BYU for many decades. And crown ethers have many applications beyond what we're showing you here, but they all are derived from the fact that they're excellent at forming these host-guest complexes with cations. Any questions? All right, so epoxides, again, they're a subset of ethers. And even though that 3-membered ring is strained, there are epoxides that exist in nature. And I've drawn one for you over here on the board. This is called paraplanone B. Many times epoxides are attached to larger rings, and this one has 2 such epoxides. This is a 10-membered ring, and then this epoxide has both of its carbons as part of the 10-membered ring. This epoxide only has one of its carbons as part of that 10-membered ring. So paraplanone B exists in nature. It is the pheromone that is secreted by the female cockroach. And in the 1970s, this was a popular molecule to study. Why would people be interested in the sex pheromones of cockroaches? What value could you get by having the sex pheromone of that a female cockroach emits? Yes. Yes, if you want to control the cockroach population, you could put this pheromone in a trap and you would attract all the male cockroaches and the cockroach population would plummet. So indeed, it's not just a chemical curiosity, it has some practical value. But there are a couple problems associated with it. Pheromones are usually emitted in very, very small quantities in nature. And so in the 70s, when they were trying to figure out the structure of this pheromone, the researchers sacrificed 75,000 female cockroaches. Okay? And I've read an account of this. Not only were they female cockroaches, I read that they are virgin female cockroaches. Now, I have no idea why they needed virgin female cockroaches. I have even less of an idea as to how they would tell that a female cockroach is a virgin female cockroach or not. But they sacrificed the 75,000 female cockroaches, and they isolated the pheromone from them, and they got 200 micrograms. That's 0.2 milligrams. That's barely enough to register on a balance. That would essentially be like a little speck of dust if you were looking at it on a balance. So they got just a teeny tiny amount of this from nature with this massive effort. And somehow they were able to use that small amount to figure out this structure, but they couldn't quite figure out the stereochemistry of the epoxides. They knew that this one had to be cis, but they didn't know if it was pointing up or pointing down. So they had 4 possible isomers of the paraplanone B. They didn't know which one was the one from nature. So along came an organic chemist who worked at Columbia University in New York City, and he decided he was going to figure this out. And so he went in the lab and he designed syntheses of the 4 different diastereomers that could have possibly been, the 4 different stereoisomers that could have been the structure of paraplanone B. And this one represented his 3rd choice, and it was the one that was identified as the actual pheromone. And how do you think he figured out it was the pheromone? Yeah, it's the one that made the male cockroaches go crazy. There were plenty of cockroaches in New York City for him to experiment on. And he found that this was the molecule that excited the male cockroaches. Okay, so how the epoxides play a role in that pheromone activity, I do not know, but indeed they're present in this naturally occurring molecule that is the female cockroach pheromone. So here we have a synthesized epoxide. This is a molecule known as ixabepilone. It is an anti-cancer drug. It is used to treat breast cancer. It's used to treat breast cancer that's resistant to other types of cancer drugs. Ixabepilone is interesting for a couple reasons. So in the terms of the structure, you see that like paraplanone B, we have a very large ring, and then we have the epoxide attached to that larger ring. Again, that's fairly common for epoxides. It's based on a natural product. There's a naturally occurring compound called epathalone B that has an oxygen here instead of this NH group. So epathalone B is an ester, whereas ixabepilone is an amide. A good friend of mine, one of my best friends, led the team at the pharmaceutical company that discovered and developed this drug. So I do know a little bit about the story. And he tells me that one of the most gratifying aspects of his job in the pharmaceutical industry is when patients whose lives have been saved, whose cancers have been cured by his drug, look him up, figure out that he was part of this team, and they contact him and thank him for this. So it can be very gratifying to work as a chemist in the pharmaceutical industry. But a lesson we learned from the development of this compound is that when you're developing a drug, you don't always want the most potent compound. So the natural product, epathalone B, the one that has an ether instead of an amide, is very potent in vitro, when you're testing it in test tubes, when you're trying to kill cancer cells in petri dishes or test tubes. Epathalone B, the natural product, is very, very good at killing cancer cells in test tubes. But then when you try to give it to animals, when you try to give it to mice or other animals, it's not very good at killing cancer in living organisms. Okay? And so my friend and his team hypothesized that the reason for that is that the compound was not stable enough to survive with all the enzymes that are present inside of living organisms. And they learned something, they knew something that we're going to learn in Chem 352M, which is that the amide functional group is more stable than the ester functional group. Now, that's something that will come in Chem 352M. But they knew this, and so they thought, well, maybe it's that ester that's causing this molecule to fall apart before it can function as a good anti-cancer drug. So why don't we change the ester to an amide? And they were very, very good synthetic organic chemists, so they knew how to do that. They knew in just 2 or 3 steps how they could swap out that oxygen for an NH group. And they did, and they found that this molecule is about 10 times less potent against killing cancer cells in test tubes. But when you give it to a living organism, such as a mouse, it's now much, much more effective. And that's because it is stable enough to survive to get to binding to its target inside the cancer cells. Okay? So when you're working in the pharmaceutical industry, it's not always the most potent compound that's going to be the best drug. It still has to have a certain level of potency, but it also has to have the right physical properties to be able to diffuse across cell membranes, survive in the cell long enough to bind to its target, and then be able to do its job. So a very interesting compound for a variety of different reasons. Any questions? All right, so let's talk now about how we prepare alcohols, ethers, and epoxides. Have we learned any reactions that could be used to synthesize alcohols? Yes, half raise of the hand there. Okay, so we could perform a substitution reaction with an alkyl halide. What would our nucleophile be if we were trying to form an alcohol? Okay, someone have the right answer? Hydroxide, yes. So let's say we wanted to make ethanol. We talked a lot about ethanol and the reasons why we would. It's useful for multiple purposes. We could take sodium hydroxide or any of our hydroxides, and we could synthesize ethanol. And we could synthesize any primary or secondary alcohol doing this. We couldn't make tertiary alcohols by this method, but so this is the reaction we know for making alcohols. The reality is it's hardly ever used, and that's because alcohols are much more abundant in nature than alkyl halides are. Alcohols are common in nature, and so usually if we want an alcohol, we could get it from nature. If it's a fairly simple alcohol, that is. And so it's more common for us to need to convert alcohols into alkyl halides. And on Wednesday, we'll learn reactions for performing that transformation. Okay, so usually we would go in the other direction, but if we need to make alcohols, this is a method we can use. But a related process to this is commonly used to synthesize ethers. And it's known as the Williamson ether synthesis. So some of the reactions in organic chemistry are named after the individuals who discovered them. Alexander Williamson was a chemist in England in the 1850s. So this reaction's been around for a long time. He was wanting to make diethyl ether, which was useful as an anesthetic and also as a good solvent. And he found that you could take an alkoxide. Alkoxides are strong bases and strong nucleophiles, just like hydroxide. So he could react an alkoxide with an alkyl halide, with iodoethane in this case, primary alkyl halide. And he could generate diethyl ether as the product. And then, of course, he's going to get a salt, potassium iodide salt byproduct. Okay? So the Williamson ether synthesis is very closely related to this reaction up here. It's simply an SN2 reaction with an alkoxide, the conjugate base of an alcohol, as our nucleophile, reacting with an alkyl halide. So this particular reaction is giving us a symmetrical ether where both of our alkyl groups are identical. if we wanted to synthesize an unsymmetrical ether with two different alkyl groups, we would have two different combinations of an alkoxide and an alkyl halide that could, in theory, be used to construct that type of ether. So the question is, would both of those methods work? And if so, would they be equally effective, or would one work better than the other? And we're going to answer that question by doing an iClicker question. So go ahead and get your iClickers out and go ahead and talk to your neighbors. We have an unsymmetrical ether that we need to synthesize. We have four choices of combinations of nucleophiles and electrophiles to generate this. And I'll partially answer the question and tell you that there is indeed one choice that's better than the others. So go ahead and talk to your neighbors and try to figure out what that is. All right, let's try to get our final answers in the next 20 seconds or so. Okay. All right, that looks like all the answers. Okay, we'll go ahead and stop that. Okay, so would we rather perform an SN2 reaction or an SN1 reaction? SN2 almost always better than SN1. So which ones does that eliminate? Which choices does that eliminate if we want to do SN2? Yeah, B and D would have weak nucleophiles, so those would only work in an SN1. So that narrows it down to A and C. Which one did you choose? Okay, there was a debate there, but I would say that A was the favorite. So someone who chose A, please raise your hand and tell us why you felt that A was the best choice. Yes, please. Okay. Okay, so the primary alkyl halide will react faster in an SN2 reaction. So the reaction of A will go faster than the reaction of C. If you're performing a reaction with a secondary alkyl halide with a species that is both a strong base and a strong nucleophile, what is the outcome typically? So you'll usually get a mixture of SN2 and E2. Okay, so that's why choice A is better than choice C. This will be faster, and it will give you the SN2 product exclusively, whereas here you will get a mixture of substitution and elimination. It is true that your nucleophile is bulkier here, so that would slow it down a little bit. So it might actually be questionable as to which would be faster because the nucleophile is bulkier here than it is here. But definitely having the primary alkyl halide would give you all substitution, whereas here with the secondary alkyl halide, you would have a mixture of substitution and elimination. So this is general. When you're needing to synthesize an unsymmetrical ether, you want to choose the pair of reagents that has the less hindered alkyl halide. You want the less hindered alkyl halide. Now I know this wouldn't work if you needed a tert-butyl group. We gave you an isopropyl group and it would work. If we gave you a tert-butyl group, that wouldn't work. You'd have to use other methods to make that kind of an ether. But it would work with any secondary alkyl group that you needed on the ether. Okay? Any questions about the Williamson ether synthesis? Yes. That's a great question, and that actually leads right into the next topic, which I was writing on the board here while you were answering that question. So the alkoxides are conjugate bases of alcohols, and so they're derived by simply treating your alcohol with a strong base. There are a variety of strong bases that will work, but the most convenient one is sodium hydride. So sodium hydride is a very strong base, and the advantage of using sodium hydride is the conjugate acid is hydrogen gas. So after that reaction occurs, the hydrogen gas byproduct just bubbles out of the reaction mixture. So it automatically separates itself from your desired alkoxide. You don't have to put any effort into separating the alkoxide from the conjugate base or conjugate acid of the base that you used. So sodium hydride is your most common strong base that is used to make alkoxides. Okay? And these are very fast reactions. We've said that acid-base reactions are rapid because a proton is very, very small. So transferring a proton from one heteroatom to another, very, very fast, very simple process. So epoxides are a type of ether. So it stands to reason that an epoxide would be synthesized using a type of Williamson ether reaction. And indeed, well, before I do that, I forgot to show you this. So because the precursor to alkoxides are alcohols, many times we would just write these two steps over a single reaction arrow to indicate that if you start with an alcohol and deprotonate it with sodium hydride in step 1, and then add in an alkyl halide in step 2, then you would get your ether product. Okay, so it's very common to combine the Williamson ether synthesis with the deprotonation of an alcohol. So as far as epoxides go, we're going to synthesize epoxides using a special version of the Williamson ether synthesis where we have our nucleophile and our leaving group in the same molecule. When that happens, we're able to perform an intramolecular SN2 reaction that's capable of forming a ring. If we want to form a 3-membered ring, we need our alcohol and our halide on adjacent carbons, and that's referred to as a vicinal halohydrin. We learned last week when we were talking about alkynes and precursors to alkynes, a vicinal means on adjacent carbons, and halohydrin just refers to a halide and a hydroxy group. So that's why this molecule is referred to as a vicinal halohydrin. I'm just going to draw in these bonds here to fill in the valences. These can either be hydrogens or alkyl groups or some combination of them. But if we treat a vicinal halohydrin with a strong base, sodium hydride is very common. Potassium tert-butoxide is probably our second most common base that's used for this process because tert-butoxide is the strongest of our alkoxide bases. We're going to deprotonate our alcohol, and that's going to give us an alkoxide. But this particular alkoxide has a leaving group on the adjacent carbon. So it's just going to perform an intramolecular SN2 reaction, generating the epoxide. Okay. And in our next chapter, chapter 10, we'll learn how vicinal halohydrins are synthesized. Okay. Any questions about the synthesis of epoxides or ethers? Okay, so let's talk now about the reactions these compounds undergo. And we'll start out with alcohols. On Friday, we looked at electrostatic potential maps of alcohols, ethers, and epoxides. And we learned that they had something in common with alkyl halides. They have an electronegative heteroatom, a very polar bond between carbon and that heteroatom. And that makes the carbon quite electrophilic. So that would lead us to believe that alcohols could undergo substitutions and eliminations in similar fashion to alkyl halides. But they cannot, as we mentioned on Friday. The OH group is a very, very poor leaving group because hydroxide is a strong base. So if we want alcohols to undergo substitutions and eliminations, we have to convert our poor leaving group into a good leaving group. There are ways to do that. We'll learn several ways to do that in this chapter. The simplest way is to use a strong acid. If we react our alcohol with a strong acid, we will protonate the oxygen, generating an oxonium ion, the conjugate acid of an alcohol. What is our leaving group in this molecule? It's water. Is that a good leaving group? It is. It's a weak base. It's a good leaving group. So now that we've converted our poor leaving group into a good leaving group, we can perform eliminations. If we have a suitable base present, we can perform substitutions if we're using a nucleophile. Okay. So that's the general strategy involved with alcohols. Yes? There could be an equilibrium there, but we'll talk about if you have a strong acid, if you have an acid, if HX is a stronger acid than this, then the equilibrium will favor this because this would be a weaker acid than HX. Okay? Depending, we'll show you how the most convenient way to do an elimination. There is a way to do it very conveniently where you don't actually have to add any reagents to the mixture. So the situation with ethers is very similar. Your leaving group would be an alkoxide instead of hydroxide, but they're of comparable basicity. They're poor leaving groups. So ethers, you're going to have to protonate as well, but to convert the alkoxide into a good leaving group. Epoxides, on the other hand, are special because of the ring strain in this molecule, and that allows epoxides to actually react with a nucleophile in an SN2 type of a process, even though your negatively charged oxygen is basic and is a poor leaving group. The release of the ring strain caused by opening up that three-membered ring provides a thermodynamic driving force that allows reactions like this to happen. And at the end of the chapter, we're going to talk about these kinds of reactions. But we're going to start with alcohols. We'll start with the reactions of alcohols. And we're going to start with an elimination reaction of alcohols that is known as dehydration. As the name suggests, in dehydration, we are eliminating water. So we start with an alcohol. That alcohol has to have at least one beta hydrogen atom. And then we're going to react that alcohol with one of two acids. We're either going to use sulfuric acid or we're going to use toluene sulfonic acid. We learned the structure of toluene sulfonic acid back in chapter 2. It's an organic version of sulfuric acid where one of the OH groups of sulfuric acid has been replaced by a benzene ring, a substituted benzene ring. And so the advantage of toluene sulfonic acid is it is soluble in organic solvents. And so oftentimes it is used in place of sulfuric acid. But when we're using alcohol, sulfuric acid dissolves reasonably well in alcohol. So we can use either of these acids pretty much interchangeably to perform dehydrations of alcohols. So we're going to generate an alkene, and then we're going to generate water as a byproduct. And then on Wednesday, we're going to learn a second way to accomplish a dehydration that does not use acid. We're going to learn that there are some drawbacks to the acid-promoted reaction. And so there's a separate set of reaction conditions using phosphorus oxychloride and pyridine that will also accomplish dehydration. So we'll get to that on Wednesday. The remainder of the time today, we will talk about the acid-promoted dehydration of alcohols. Okay? So there's some data we need to discuss about this reaction, and then we will describe a mechanism for this reaction that's consistent with that data. Okay? So relative rates. What kind of alcohols react faster than others? In dehydration reactions, our primary alcohols will react the slowest. Secondary alcohols will react faster, and tertiary alcohols will react the fastest. So more substituted alcohols are going to react faster in a dehydration reaction. Okay? And then, as was the case with dehydrohalogenation, with some dehydrations, we have the possibility of generating two or more different constitutional isomers. So if we take this particular alcohol, this is 2-methyl-2-butanol, and we react that with sulfuric acid, we have two possible constitutional isomers that could form. Okay. Of course, we're going to make water as well. And according to Zaitsev's rule, the more substituted alkene would be the major product. And in fact, dehydration, like dehydrohalogenation, does follow Zaitsev's rule. So the more substituted alkene will be the major product. The less substituted alkene will be the minor product. With dehydrohalogenation, we also had stereoselective reactions. We had eliminations that could produce cis or trans isomers, and we said that the trans isomers would be favored. That is going to be true in dehydration as well. Dehydrations will also be stereoselective and will give a trans alkene as a major product. Okay? So we need to come up with a mechanism that will explain, that will be consistent with the data that we have here on the board. Now, the tricky thing about dehydrations is the mechanism is going to differ depending on the type of alcohol you are using. If we have a secondary or tertiary alcohol, it is going to be an E1 mechanism. So we'll draw that one first. Let's start with the tertiary alcohol, tert-butyl alcohol. Okay. But this is going to be a little bit different than the E1 reaction we learned in chapter 8, because we have a poor leaving group. So our first step, I'm just going to draw out part of the sulfuric acid structure, is going to be protonation of our alcohol. Okay? That's going to give us an oxonium ion, the conjugate acid of an alcohol, and that's going to be very fast. All we're doing is transferring a proton from one oxygen to another. That's going to occur very, very rapidly. Okay? So based on what you know of the E1 mechanism, what do you think will happen once we have this oxonium ion? What will our next step be? Hopefully we still remember the E1 mechanism. Go ahead. The leaving group will leave, yes. So we're going to lose water. And when we lose water, what is left? A carbocation. Okay? So we have a reactive intermediate, a carbocation. We know this step is going to be slow. All we're doing is breaking a bond in order to get to the carbocation. No bond formation. Okay? All right, so now once we have our carbocation, we're going to use our conjugate base, HSO4 minus, which I'm going to draw out in this fashion, just drawing out one of our negatively charged oxygens. That's going to deprotonate one of our beta hydrogens of our carbocation. That's going to give us our alkene product, and it's going to regenerate our sulfuric acid. So dehydrations are actually catalytic in acid. The acid gets regenerated in the second step. Okay? So this is an E1 reaction, but it has one additional step necessitated by the fact that we have a poor leaving group in our starting material. We first have to protonate that poor leaving group, convert it into a good leaving group before it will leave, giving us the carbocation. Now, the reason we need to use sulfuric acid or toluene sulfonic acid is that the conjugate bases of these acids are weak nucleophiles. You'll say, wait a minute, they're negatively charged. Well, we're elaborating on things we learned in chapter 7, and now we're learning that indeed there are some negatively charged species that will be weak nucleophiles. And what makes sulfuric acid, or the conjugate base of sulfuric acid, a weak nucleophile is that negative charge can be delocalized by resonance onto two other oxygens. That negative charge is shared by 3 oxygens. Okay? And so that resonance delocalization makes it weakly nucleophilic. It's a weak base as well, but it's better as a base than as a nucleophile. So with sulfuric acid or toluene sulfonic acid, with a very similar structure, the conjugate base of our acid will perform an elimination. As we will learn on Wednesday, if we were to use HX, one of our halide, hydrogen halide bases, then we would see substitution as opposed to elimination. So if we want to perform a dehydration, we need to use one of these 2 acids in the process. Okay? And so we know that more substituted alcohols, oxoniums, we learned it with alkyl halides last week, but we know that the more substituted species will react faster in an E1 reaction because we'll form the carbocation faster. But something different has to be happening with a primary alcohol. The primary alcohol cannot react via an E1 mechanism. Why not? We can't form the carbocation. The carbocation would be too high in energy. So something else has to happen in order for dehydration with a primary alcohol to occur. So we're going to start out in the same fashion and protonate. Here I've used toluene sulfonic acid and just drawn the abbreviation. Okay, so now we have our oxonium ion, but we're not going to form a carbocation. Instead, we're going to perform an E2 process where the conjugate base, the toluene sulfonate anion, is going to deprotonate, and that's going to give us our alkene. That's going to release water, and that will give us our toluene sulfonic acid back. Okay? So this is an E2 reaction. You might wonder, well, why is it slower than the E1? Because we said primary alcohols react slower than secondary and tertiary alcohols. The reason is this tosylate anion, this conjugate base of toluene sulfonic acid, is a weak base. Okay? So we've told you in chapter 8 that you have to have a strong base for E2. We're modifying that a little bit here, as we often do in organic chemistry. You can have E2 in some circumstances with a weak base, but it's going to be pretty slow. In this case, even slower than an E1 reaction. Okay? So because in these dehydrations we're using weak bases, the conjugate bases of strong acids, then the E2 reaction is going to be really, really slow. We're only going to see it in the case where we can't form the carbocation. We're actually going to form carbocations faster than we would do in E2 in the cases where carbocations can be formed. Okay? So these two mechanisms, E1 for secondary and tertiary alkyl halides, E2, or alcohols, sorry, E2 for primary alcohols, explain the relative rates, which are that the more substituted alcohols react faster. Another feature we see from these mechanisms is that they're catalytic in the acid. The acid is regenerated, and so you can use a catalytic amount of that acid. Questions? So let's look at the thermodynamics of the dehydration reaction. The bonds, the bond dissociation energies of the bonds that are formed and the bonds that are broken. We're forming a pi bond and a hydrogen-oxygen bond, and we're breaking two sigma bonds. Knowing that pi bonds are generally weaker than sigma bonds, we would expect this reaction to be endothermic, and in fact, it is endothermic. Okay. So the dehydration is endothermic. This equilibrium favors the alcohol and not the products. But there is a way to manipulate the equilibrium to drive it towards formation of the products. How would we do that? Think about what you know about Le Chatelier's principle. Yes? Okay, increasing the temperature will help. So you know that, so heat is a reactant when something is endothermic. There's a specific reason why increasing the temperature helps in this case. Any ideas on that? Yes? Okay, so if you add a reactant, it shifts to form the products. That doesn't really work with heat, though, in this case. Yes? Okay. So if you can remove one of the products from the reaction mixture, then the equilibrium will shift to make more of the products. And that's exactly how we manipulate the equilibrium to favor the alkene product. So the alkene is always going to have a lower boiling point than the alcohol it is derived from. Alcohols can hydrogen bond to each other. Alkenes cannot. So you can always heat the reaction up enough so that this is distilling off, and then you can collect it somewhere else as it forms, and then more of it will form. If you have a longer chain molecule, you might get to the point where your alkene has a higher boiling point than water. So then you just distill off the water. One of these two products is always going to have a lower boiling point than your alcohol starting material. So you can always choose a temperature that will allow you to distill or boil off one of the two products and shift the equilibrium to favor the products. Okay? You might also notice this is a type of fragmentation. You have one starting material and two products, so that's going to be favored by entropy, but not enough to overcome the enthalpy problems. Okay? So, last thing we'll do today is we'll look at a dehydration of this particular alcohol. So I've drawn this alcohol. We have a quaternary carbon here, so there are no beta hydrogens on that carbon, but we have beta hydrogens here. So how many possible dehydration products would we have in this case? We would have one. So if we treat this with sulfuric acid or toluene sulfonic acid, we would expect this to be the only product that we get. However, we get a very small amount of this material. We get about 3% of that. It is formed, but it is formed as a minor product. The major product looks like this. We get 64% of this compound. And then the minor product is the less substituted version with the same skeleton. We get 33% of this one, okay? So something unusual is going on. If you look at these two products that are formed in much more abundance than the one we expect, you see that we've had a rearrangement of the skeleton. And so what we learned from this reaction is that certain carbocation intermediates can undergo rearrangements to give more stable carbocations. So we'll talk about the mechanism of that and how that process occurs on Wednesday.